Hello, my name is Eva Elviani, and today I will start our tutorial with the introduction to Galaxy for Omics integration. So, what is Galaxy? Galaxy is an open source platform designed to simplify data analysis in computational biology. Its user-friendly interface makes it accessible to researchers even without programming expertise, and it supports various types of data including genomics, proteomics, and metabolomics. So, why omics integration matters? So, omics integration is essential because it allows us to combine datasets from different biological layers such as DNA, proteins, and metabolites. This holistic approach helps us uncover complex interactions and provides deeper insights into biological systems. So in this tutorial, we aim to demonstrate how to use Galaxy for integrating datasets, analyzing results, and interpreting findings, all while highlighting its features and limitations.
Welcome to our tutorial on using Galaxy for omics data integration. Galaxy is a versatile platform that can be accessed in many ways. The easiest option is to use the public web-based version, where no installation is needed. The other way is through local or cloud deployment. For this tutorial, we will be using the online platform. Now let's look at the tutorial. First, let's access Galaxy. Simply open your web browser and go to usegalaxy.org. To fully utilize Galaxy's features, you will need to create a free account. Here's how. First, click on the Login or Register button at the top right corner of the page. Select Register and fill in your email, username, and password. The last step is to verify your email address to activate the account. Once you are logged in, take a moment to explore the interface. On the left, you will find the tool panel with a variety of analysis tools. The middle section is the workflow canvas where you will build and run analysis. On the right, you will see the history panel which tracks your uploaded datasets and analysis steps. Now let's move on to acquiring datasets. Acquiring the right datasets is crucial for omics data integration. The first one is through Galaxy Shared Data. Galaxy itself offers public datasets through the Shared Data tab. You can explore data libraries or import datasets from published histories directly into the workspace. For online sources, we can use GeoGene Expression Omnibus, where it offers transcriptomics data in CSV format. Next is EBI Array Express. It provides proteomics and transcriptomics datasets. And last one is Metabolites, which is great for metabolomics data. Normalization is an essential step to ensure data from different sources or scales is comparable. It helps remove biases introduced during experiments or data collection. There are several tools that can be used for normalization. The first one is the Galaxy itself. In Galaxy, tools like Normalize and Data Mesh can clean and scale data effectively. The next one is using spreadsheet software like Excel or Google Sheets. The common methods for normalization include min-max scaling, Z-score normalization, and log transformation. These tools and methods ensure that the data is ready for integration and further analysis.
Thank you for the explanation on the data acquisition and normalization, Priska. Now I, Elna Elisepson, will continue with the workflow of the omics. Before integrating all three of the omics that we have chosen, it is important to process the data beforehand. For the first omic, I will talk about proteomics workflow. After we acquired the data and normalize it, we need to quantify the proteins using several methods, which are label-free quantification, isotope labeling quantification, and normalization. And the second step would be post-translational modification analysis. For label-free quantification, it is used to estimate the protein abundance by comparing protein intensities between samples. This will use MaxQuant and MSStats tool. The next one is isotope labeling quantification. It is used to derive the quantitative info from labeled proteomics using the labeled to unlabeled peptide intensities across different samples. This method will use the MS Stats tool. And the last one is the normalization, which is used to normalize data to correct some systematic error, which also use the MS Stats tool. And lastly, the post-translational modification analysis of the protein can be done when we want to identify modification like phosphorylation, acetylation, or glycosylation that regulate the protein function. To do the PTM, we can use the MaxQuant tool in Galaxy. Next, I will talk about the transcriptomic workflow, which involves 4 steps. The first one is the quality control. The second one is the align reads to the reference genome. The third one is the quantification, and the last one is the normalization. For the transcriptomic data, after acquiring it, we need to do a quality control to guarantee the accuracy and dependability of the collected data. In this part, tools such as FastQC and Trimomatic are used to cut the low-quality bases. Then, we need to align reads to the reference genome using HISAT2 tools to read it in BAM format. BAM format uses a specialized compression algorithm for indexing and record retrieval and are not human-readable. After that, by using string type tool, we can perform quantification on the transcriptomic data to measure the abundance of transcripts. Lastly, by using the DSEC2 tool, normalization can be performed to normalize the row counts in the data. For the metabolomic workflow, the first step is to determining the metabolite peaks. After normalizing the data, we can determine the metabolite peaks based on the raw spectral data that we have obtained. To do this, we need to use the XCMS tool, which helps in detecting the peaks across the data. Next, peak matching was done and applied to individual samples, as this will allow for comparison of samples across peak intensities and allows for correction of retention time deviation. To do this, we need to also use an XCMS tool, and we will obtain the aligned peak list. To correct the retention time, XCMS tool is used by aligning retention time of all samples in single step to ensure consistency. Then we have to fill in the missing peak data. We can determine which samples are missing from each peak group when there are no peak detected for those samples. We could use the information obtained from peak detection about where peaks begin and end, and align retention times for each samples. Then use peak list from XCMS to fill in the missing peak. Lastly, normalization was done to adjust the drift in intensities. We can do this by using the Match MS filtering tool in Galaxy. That's all from me. Now for the integration part, I will pass to Valerie to explain more about it.
Integrating different omics using Galaxy integration platform can be simplified into 4 steps, which are acquiring data from public source. Step 2, upload datasets into Galaxy History. Step 3, process datasets using specialized tools. And finally, export datasets and interpret output in report form. To import the acquired datasets, click on the upload symbol at the left panel. Drop files or choose local file to search for its location. Click to start for saving it to history section. Next is further explanation about the tools interface available.
The data acquired need to be uploaded into Galaxy Integration Platform for processing processes. Galaxy Integration Platform contains a lot of functional tools to help with the process. This tool can be found at the left panel. The tool search helps in finding a specific tool in a crowded toolbox by entering name of the tool. For example, HISAT2 for a fast-sensitive alignment reads of a reference genome. Click on the results. In the running tools interface, they found to be 9 functional keys and info such as input parameters, input dataset files. Make sure the uploaded file just now is in the correct format, and Run to button to execute dataset outputs. Help information and tool citation can be found at the bottom of the tool interface. Next, on the top corner of the tool interface, they found a star symbol if user wish to add tools to a favorite for easy search in future. And then, a new tool version can be installed without removing the old one to ensure reproducibility. Other options, and lastly, save file location.
Here are some examples of integration tools for different omics types such as transcriptomics, proteomics, and metabolomics at each stage of their workflows. The use of multiple tools and the overlap of tools across different workflows in omics integration depend on factors such as dataset file types, the needs for researchers to optimize workflow outcomes, or the fact that many tools perform similar tasks but differ in algorithms, sensitivity, specificity, or intended application. Additionally, some tools are commonly used across various omics workflows.
Hello, my name is Shania and I will be explaining about the key features which are divided into 6 parts. So the first part is data integration, where it integrates multiple omic data sets to identify relationships, and the tools include Diablo, SPLSDA, and Clustered Image Map (CIM). Moving on to variable selection, it identifies key variables driving dataset relationships using sparsity-based methods, and the tools include SPLS and SCCA. Next, dimension reduction projects high-dimensional data into lower-dimensional spaces, and it comprises 2 plots, which are sample plots and variable plots. For multi-block analysis, it integrates more than 2 datasets to highlight shared and unique features, and the tools include Diablo Workflow. Visualization tools, on the other hand, create publication-ready plots like correlation circle plots, heat maps, and network plots. Lastly, biomarker discovery pinpoints important variables contributing to dataset relationships, and the tools include SPLS, DF for biomarker.
Next, I will present the limitation for Galaxy integration tool. There are 2 limitations that are most common, and the first one is technical limitation. Technical limitation can also occur when combining diverse omics. This is because they have different forms and properties. Missing value are also a type of limitation, but it can be fixed. There will be difficulties when interpreting data with several omic layers. Without clear instructions, user may encounter difficulties understanding complex omics between various omics datasets. Galaxy software also does not have common processing and analyzing data from different omic types. Therefore, the difference may result in different level of resolution and analytical techniques, making the process more difficult.
To conclude, this tutorial demonstrated the use of Galaxy as a tool for omics integration, showcasing its ability to combine genomics, proteomics, and metabolomics data for holistic analysis. So Galaxy offers a user-friendly interface, scalability, and compatibility with diverse data types, making it accessible and versatile for researchers. Apart from that, alternative tools such as Mixomics, MOFA or MOFA, and iCluster Plus can also be considered for omics integration depending on specific research needs. While Galaxy requires some familiarity with bioinformatics, it remains a powerful platform for integrating and analyzing complex datasets. Users are heavily encouraged to explore its features further and apply them in their research.